All right, the first section, Introduction to the Cell, we'll be talking about microscopes. So the microscope, the light microscope, is the first, I guess, most primitive type of microscope. We're using beams of light as well as curved glass or mirrors to magnify an object. Specimens can be magnified by up to 1,000 times, which is a relatively huge limitation for small specimens. However, you can view specimens that are alive and view how their cells or how their structures are moving while they're alive. With regard to light microscopy, we have to be concerned with magnification and resolution. So as we increase the magnification, the resolution becomes less clear, as you can see in the picture to the right of the dog and the dog's nose. So as one magnifies, we have to also make sure we're modifying our scope using our fine adjustments to make sure that our magnified specimen has a clear picture of it. All right, this is a nice illustration of a root hair, cross section of a root hair, and how you can see those individual cells of this particular specimen. And again, those cells, if we were to magnify them, there's internal structures to them which we cannot see with the light microscope. So just kind of highlighting those limitations. These microscopes have been utilized to study microorganisms, animal and plant cells, bacterial cells, and even structures within cells. And these early tools and instruments helped to develop something called the cell theory. This is this concept that all living things are composed of cells. The cell is the smallest package that life can come in, and that all cells come from pre-existing cells, and we are examples or living proof of that. We are the result of our parent cells coming together to create a brand new unique cell. So that is what is meant by that pre-existing cell. What's curious, however, is the question, where did the first cell come from? There's a couple of different theories that that have suggestions of what that answer might be, but again, there's no hard evidence to support that being correct. All right. These next three pictures are going to illustrate a paramecium under different forms of microscopy. This is a light microscope, a micrograph, a photograph, a microscope photograph. And as you can see, the edges here, there's no better way to describe it. It almost looks kind of like a fine fur compared to this image, which is an electron micrograph. And this is what is the difference between the resolution and the tail. You can see the fine cilia that cover this entire creature's outer surface versus, it's just kind of a blur. The light's actually going right through this, so you can't even see, the front-facing surface. Right. This is an example of a transition electron micrograph where a beam of electrons are sent through the cell, just go right below the surface, the external surface, and bounce back to give us a picture of its internal anatomy. Right, and this is an overlay of a scanning and transmission. And here you can see kind of giving a full picture of this specimen. It helps understand how it lives and works and interacts in its world. These graphics just illustrate, how the range in which these tools allow us to view. So the unaided eye down to the human egg cell, which almost looks like a little grain of pepper. All right. And then with the light microscope, and the parts that I find most interesting are the ends right here. So we can see individual cells. We can even view bacterial cells with light microscopes. However, the resolution is very poor. So electron microscopes change how we see that, and their range even has limitations at the molecule and atomic level. All right. This is just illustrating kind of a graphic, and here's our scanning and transmission, just definitions for them. Scanning is providing the outer surface, versus transmission is going into the innermost anatomical pieces.